All right, it's my pleasure to introduce Sujita Balami. She was born and raised in Nepal. Her research interests are aquaculture, fish pathology, and molecular biology. She earned an undergraduate degree in Nepal and a master's degree in Vietnam. And she's currently a PhD student working with Dr. Matt Griffin and Dr. Larry Hansen at Mississippi State. Her dissertation work focuses on the development of Edwards-Yella pesticide vaccines in catfish aquaculture. But today she's going to discuss an interesting case of streptococcus in farm-raised catfish. So thank you very much for being here, Jimmy.
Thank you, Dr. Phelps. So, hey everyone, I'm Sujita for the people who don't know me. So I'm from Nepal and currently I'm a second year PhD student with Dr. Matt Griffin. And my actual project is that I'm working on developing a killed and live attenuated vaccine for Edwards-Yella pesticide. But today I'm not going to talk about it. So today I'm gonna talk about a case of genetic identification of new fish-associated mortality that is caused by gram-positive bacteria in catfish. So let me start. So what's the problem? In June 2022, a 10-pound male channel catfish was submitted for aquatic diagnosis at the Aquatic Diagnostic Laboratory, Stoneville, Mississippi. And there were significant lesions, pendulous abdomen was seen, and red cell vent was there, and the acidic fluid flowed from the abdomen when cut open. And the cultures that was made in Miller-Hinton agar with 5% sheep's blood showed small white non-hemolytic colonies. A routine diagnostic workup was done. Cytological examination of the acidic fluid was done, which revealed numerous extra and intercellular coccoid bacteria on staining. And there were numerous individual bacteria, numerous individual bacteria as in here, and some were found in chains too. These are the histopathology slides obtained from Aquatic Diagnostic Laboratory done by Dr. Lester Koo. These are the kidney and testes, liver, and spleen of the fish. And the most common abnormality that you can see in these histological slides are the thickened capsular surface in all the organs. So I'm going to show you all the organs one by one. So the kidney was diffuse and it had interstitial nephritis, which increased towards the thickened capsular surface here. And there were hemorrhages around the tissue and some macrophage aggregates that was inside the tissue. Now moving on to the liver. Liver showed myosericitis, that is inflammation around the cellular surface and thickened capsular surface. The testes showed myosericitis and inflammation within the surface. As you can see here, small hemorrhage. And the spleen had splenitis with erythroid depletion and increased number of pigmented macrophage aggregates as in the kidney. As you can see here, a lot of macrophage aggregates that can be found. And the most common abnormality, as I said before, was the thickened capsular surface. See how big it has gotten, and there were hemorrhages inside the capsular surface as well as the fused aggregate of macrophages. All this histopathological presence of the bacteria and inflammation, it indicates that the bacteria might have gotten into the fish through the vent and it's making its ways inside the tissue to infect the fish. So for the genetic identification of the bacteria that caused this inflammations, 16S rRNA sequencing was done the bacteria with DNA extraction using the PerioSpin DNA isolation kit. PCR was done using fusion green measurements, and amplicon purification was done from the PCR product, and DNA sequencing was done at Eurofins Genomics. So after the results came in, a blast search was done in NCBI database, and it showed that the gram-positive cocci that was found inside the tissues of the fish were Streptococcus dysgalactiae. But the 16S rRNA only doesn't necessarily identify the bacteria into the species and subspecies level. So this warrants a further investigation regarding the identification of the bacteria. So now let me talk a little bit about Streptococcus species. So Streptococcus is a gram-positive bacteria, and it appears usually long chains in the growth media. It is associated with cutaneous lesions, cardiopulmonary disease, or adenoiditis in humans. In horses, it is usually considered as a very normal part of the microbiota in the skin and mucous surfaces, and it has been isolated from infections of the horse reproductive system, upper respiratory tract infections, including case of Stangl-like disease and death of the horses. And lastly, it has also been found in cow or cattle, which cause mastitis and subcutaneous lesions. So when I was doing my research, I found that there is a typical classification named as Lansfield classification that is done to classify the catalase negative gram-positive cocci based on the carbohydrate antigens that are found on their cell walls. And all the fish isolates that has been found till now has been grouped in Group C, and are called Group C Streptococcus dysgalactiae, or it was abbreviated as Group C Streptococcus dysgalactiae, GCSD. So moving on to the fish research now, what has the bacteria caused in the fish? So the first case of Streptococcus infection was reported in rainbow trout by Bookmer et al. In 1979, but it was not Group C, but it was grouped as Group D, and they were not identified genetically at that time. The most common symptoms that were seen in the rainbow trout were exophthalmia, splenomegaly, and hemorrhage in the liver with discoloration. Then there were other recent reports of the bacteria in the mortality of fish. So the second example is from the Nomoto et al. 2004, where it was identified that the amberjack and yellowtail were having caudal peduncle lesions and mortality, and it was associated with Streptococcus dysgalactiae. Lau et al. Also found similar lesions, exophthalmia and ascites in the abdominal cavity of Nile tilapia in Taiwan, whereas in the US in 2021, Hawk et al. Found the hemorrhage near the eye and operculum and head in the silver carp. There has also been other reports of this bacteria causing disease and mortality in Nile tilapia in the US, as well as a new case has been found in China for the death in the channel catfish associated with the Streptococcus dysgalactiae. So, as I started reading some more papers, I found that it has not only been classified as Streptococcus dysgalactiae, but it also has been grouped into subspecies. And there are two subspecies, that is Streptococcus dysgalactiae, subspecies dysgalactiae, which is abbreviated as JSD, and Streptococcus dysgalactiae, subspecies equisimilis, STSE. And this is a very recent research that has been published by Shoemaker et al. 2022, where they reported the bacteria in Nile tilapia. And you can see that the fish clades in the previous study has shown that they are distinct from horses and human clades, and they form their own group despite of all the other humans and horses and cattle group. So for further identification, we use recently developed multi-locus sequence analysis, MLSA, which was developed by Hickman et al. And we did the same process as we did for 16S rRNA and extracted the DNA, and we did the PCR for all the 9 hash-skipping genes with using the primers indicated here and sent them for sequencing at Eurofins. And after the results came in, we did a blast search, and all the 9 genes indicated here showed a result of indicating that the bacteria was indeed Streptococcus dysgalactiae. Now, after this identification, we found some of the papers that was done in investigating the Streptococcus dysgalactiae in freshwater fishes like Night tilapia. And Dr. Griffin contacted some of the collaborators, and we were provided with some more isolates, as indicated here, from tilapia. These isolates indicated here, USDA, was from Aquatic Animal Research Unit, Auburn, Alabama, and the LADLs are from Louisiana Aquatic Diagnostic Laboratory, Louisiana. And these isolates were all identified as Streptococcus or were suspected to be Streptococcus. So we did a whole genome sequencing of all those isolates. We extracted the DNA using Biogen kit, and the library preparation was done using Oxford Nanopore native barcoding kit, and the sequencing was done using Nanopore Gridiron, and the assembly was done by Dr. Caitlin Older from USDA. So the genome sequencing was done both with the short read Illumina sequencing and long read nanopore sequencing. And this is the whole genome that has been annotated with the genes that we used for our identification. So after the whole genome was sequenced, all the isolates that were obtained, all the isolates that were obtained in our study and that was obtained from the blast hits, we formed a local database of the 51 isolates and used it to build a phylogenetic tree. So the tree shown here was built in Mega 11 after the concatenation of the 9 housekeeping genes. I'm sorry, it looks kind of messy in here, but I'll try to explain to you. So this tree revealed that the top portion over here are all the fish isolates, and they are all 100% identical. And other animal, human, and cattle isolates are down here, and they are not 100% identical to the fish isolates. So we also did average nucleotide identity calculation and digital DNA hybridization calculations to identify our species or identify our bacteria into species and subspecies label. So when our bacteria was compared with aspiogenes, it was found that the ANI was less than 87% and the DNA hybridization was less than 70%. Whereas when compared to the subspecies equisymbols, it was found that the ANI was 95 to 96% and digital DNA hybridization was 71 to 79%. And the next calculation was done for the subspecies dysgalactiae with other human and animal isolates. The ANI was 97 to 99% and DNA hybridization values of 81 to 99%. But when all the fish isolates were compared here, the fish isolates from tilapia, silver carp, amberjack, and catfish, it was seen that the ANI was greater than 99% and the DDH was 95 to 99%. This indicates that the Streptococcus dysgalactiae, subspecies dysgalactiae, is found in the fish, they are within the same subspecies as other animals, but they form their own discrete clade that is distinct from human and animal isolates. So there has been other studies and are well documented about the identification of the bacteria using the SORD gene. As we had the whole genome, we compared the whole genome with the SORD sequence across all the isolates used in this study and found that the fish isolates in the study are 100% identical across 425 base pairs of SORD sequence, and US isolates share 99.8 to 100% identity at SORD to fish isolates from Japan, China, Malaysia, and India, reported by Nomoto et al. 2007 and Abdel Salam et al. 2010. And fish isolates share greater than 99.5% identity to other Streptococcus dysgalactiae subspecies dysgalactiae from terrestrial animals and humans. This provides a further evidence that the fish isolates from their own discrete lineage. So this image is just an ongoing investigation. This is called a blast ring image generator, where our reference genome is compared with other genomes, and the innermost genome that I use here is my US isolates, and the blue circles, those are all fish isolates. But within the fish isolates, you can see that there are some regions that are absent in fish isolates, but are present in the US isolate. So this is just an ongoing investigation, but we can see that there are some unique divergent regions in US cactus isolates, which are not present in other isolates. So it might be anything like a phage element or some insertions into the genome from any source. But we are trying to do further comparative genomics to identify each genomic elements and find out the differences. This help is done by Dr. Hassan Tekeder. I would like to thank him for this support. And now, for the conclusions, this study report the first incidence of cactus mortality associated with Streptococcus dysgalactiae, subspecies dysgalactiae, and phylogenomic analysis indicate that the fish-associated streps are a unique subgroup within the streps from human and animals. This, coupled with other recent reports of STSD, and suggests that it may be an emerging pathogen in United States aquaculture. Further investigations are, however, required to assess the risk of this potential emerging pathogen. So in the end, I would like to thank my funding and funding agencies and the support who has made this project possible. I would like to thank Dr. Griffin and Dr. Lestico for providing me this opportunity to work on this project and present this. I would like to thank Dr. Caitlin Older for all the sequencing help, and Dr. Hassan Tekeder for the genomic analysis. So now I would like to take any questions, and this is the email. If you have any further questions that cannot be discussed here, you can email me. Thank you.
Great, thank you. Interesting case and good job with it. Any
Thank you,
questions
Dr.
there? Feel free to put them in the chat or just unmute yourself.
Hey, Sujita, I'll chime in. Great job. Excellent summary and excellent work. Just out of curiosity, have you given any thought to why we're seeing this now? So this was something that was an issue in Asia in the early 2000s, and now it just seems to be showing up in the US. Do you have any theories on what might be, why we haven't seen it before, and why are we seeing it now?
I don't know actually what might be the reason, but I can say that like maybe US is a huge importer of fishes from Asian countries, so it might have traveled through when importing the fish from other countries.
probably not a bad thought.
All right, any other questions out there? Well, you're getting compliments in the chat, Sujita, and a virtual round of applause to you. Good job.
Thank you, Dr. Phelps.